Hi folks, my name is Cole, and I have a Master's of Immunology. Today on Investigate, Explore, Discover, we're going to be looking at the interplay between alcohol, osteoporosis, the gut microbiome, and age. So hang around with me throughout this entire video to get all of the relevant background information so that way we can dive into some exciting experimental results. Plus, if you look in the description below, there's some information on how to get your hands on a free NFT. Now, to start off, we're going to talk about osteoporosis. Osteoporosis is becoming a greater concern worldwide as more and more people are living longer. By 2050, it is estimated that 2.1 billion people, just over 20% of the world's projected population, will be over 60 years old. Aging populations present a unique set of challenges because typically, as people age, they require some form of care. And as more and more of the population requires that care, there is subsequently less of that population to give them that care. Already, globally, in people over just 50, 1 out of every 3 women and 1 out of every 5 men will suffer from an osteoporosis-related fracture, which says nothing about how many people actually have osteoporosis and just don't know it yet. Annually, there are over 8.9 million osteoporotic fractures, which means that there is 1 every 3 seconds. When focusing on just hip fractures, in 1990, there were 1.7 million. This number is projected to increase to 6.3 million by 2050. Now, this is no trivial problem, as after experiencing osteoporotic fractures, 40% of people cannot live independently, and 80% of them cannot execute basic activities such as shopping independently. Bone is a living tissue that is constantly being broken down and replaced, much like every other tissue in our body. Osteoporosis occurs when the creation of new bone doesn't keep up with the loss of old bone. As a result, bones become weak and may break from a fall or, in serious cases, from sneezing or minor bumps. Osteoporotic bone breaks are more likely to occur in the hip, spine, or wrist, but other bones can break as well. So as you can see here in this diagram, there's decreased density of the bone in osteoporosis. Not only does this cause bones to break easier, it can also cause decreased height and stooped posture. Our bone formation is mediated by cells in our body that can create and break down bone. The cells that break down bone are called osteoclasts, and the cells that make bones are called osteoblasts. Now, when the activities of osteoclasts and osteoblasts are balanced, bone formation occurs normally. When the balance shifts towards more bone resorption than bone formation, or an increase in the cells associated with it, this leads to osteoporosis. Now, this dynamic balance between the osteoclasts and osteoblasts can be altered by many things. There are internal risk factors associated with osteoporosis, such as your age, your race, sex, or having a family history. And there are external factors, or things that you do, such as smoking, not getting enough calcium or vitamin D, taking certain medications, and excessive alcohol intake. The alcohol intake is of a bit greater concern because worldwide, lots of people drink alcohol, and I can say that I contribute to that as well. However, alcohol is known to have direct toxic effects on bone cells, notably changing the activity and proliferation of osteoblasts. It also alters bone and mineral metabolism. Now, the number of people affected by alcohol-induced osteoporosis is gradually increasing and is commonly being seen in clinical practice. Alcohol affects not only the bone, but many other parts of our bodies as well, from the nervous system to our immune system to even our gut microbiome. Now, our gut microbiome is involved in maintaining processes all over our body. It has been shown to help regulate anxiety and depression, our ability to digest the things that we eat, it can modulate our immune responses, and it can even affect the regulation of bone metabolism. Now, the gut does this by maintaining populations of healthy gut bacteria like Bacteroidetes, Firmicutes, Actinobacteria, and Proteobacteria, which all interact with each other to facilitate the gut being able to regulate other bodily systems. Now, when exposed to alcohol, this causes the opening up of tight junctions in the gut, allowing bacteria to penetrate the protective barrier, which causes bacterial overgrowth of pathogenic bacteria, reducing their clearance. And this disbalance of bacteria reduces antimicrobial compounds being formed by healthy gut bacteria and activates the immune system. Our immune system works in an interconnected fashion throughout our bodies to help clear infections or disease. We have two major arms of our immune systems. They are the innate immune system, which is fast-acting and non-specific, and the adaptive immune system, which is slow to initiate the first time, but gives a tailored and specific response to infections, and is fast-acting upon second exposure. Now, there's one set of cells which are part of our adaptive immune response called T cells that help to react to infection and stimulate other cells. In particular, CD4 T cells can differentiate into many different cell types. Two of them they can differentiate into are Th17 cells, which are involved in autoimmunity, and T regulatory cells, which are involved in tolerance. The interplay between these cell types and their populations play crucial parts in balancing the immune system and other systems in our body appropriately. One of these systems that these immune cells help regulate is osteoclast and osteoblast function and proliferation through the release of chemical messengers called cytokines. Now, osteoclasts in particular proliferate when cytokines like RANK-L, TNF-alpha, and MCSF are produced around them. These cytokines can be secreted from T cells through NF-kappa B activation. Now, I want to take a moment and just highlight why research on the topic of osteoporosis and the things that affect it is important. It is important because consuming alcohol is a risk factor in osteoporosis, which affects many older people per year. And maintenance of the gut microbiome is important for our health and is affected by many factors like alcohol consumption and age. The relationship between alcohol-induced osteoporosis and the gut microbiota in different ages has yet to be explored. Now, if you also think that these are some important reasons to research this topic, go ahead and tap the like button. This brings us to the paper that we are focusing on today, called Gut Microbiota is Involved in Alcohol-Induced Osteoporosis in Young and Old Rats Through Immune Regulation, by Cheng et al. From the Jinyu District People's Hospital of Chengdu, China. And in this paper, the authors look at old and young rats exposed to long-term drinking to establish their alcohol metabolism ability, bone characteristics, and gut microbiota composition. So to start out, the authors used young and old rats, aged 8 and 72 weeks respectively. And we're going to differentiate them by having the young ones wear hip hats and the old rats being more gray. The authors exposed these rats to 5% alcohol diets over 12 weeks, or didn't, to compare what happens to rats that drink or do not drink. So when examining these different treatments and measuring the levels of alcohol dehydrogenase and acetaldehyde dehydrogenase, which indicates how well they can process alcohol, and comparing the enzyme levels to older rats, the authors found that older rats had decreased levels of alcohol dehydrogenase and acetaldehyde dehydrogenase. Thus, older rats had a decreased ability to process alcohol. This doesn't mean that they get drunk faster, it just means they have an intolerance to it, which is way less fun. The authors next verified how alcohol consumption then affects the rat's bones. So the authors found that in young rats exposed to long-term drinking, there was decreased signs of healthy bone and increased osteoclasts and their activation signaling pathways. In old rats, both the signaling used to induce osteoclasts and their numbers were also increased. So to further verify the effects of alcohol on bone resorption and formation in rats of different ages, the authors asked, how does alcohol influence biochemicals in the blood? So to do that, they looked at serum biochemical parameters and found that alcohol increases osteoclast marker TRAC-P5 alongside modifying other biochemicals that relate to decreased bone density. When comparing young rats to old rats that did not drink, they found a gross decrease in bone density metabolites, indicating that bone density just decreases as you age. And when comparing young rats to old rats that did drink, they found increased bone degradation signals, which means that alcohol is really exacerbating this. Now, with all of this going on in the blood, the authors inquired about what exactly is going on in the bones. What role is the immune system playing in contributing to osteoporosis? Because many immune cells are present throughout our bodies, and many immune cells also start developing in the bone marrow. In the bones, they found that across age groups, alcohol increased the number of CD4 T cells, alongside increasing the number of autoinflammatory Th17 cells, and increasing the levels of suppressive T regulatory cells. They additionally found that there was an increase in pro-inflammatory cytokines present in older mice that were drinking long-term, such as TNF-alpha and IL-17, and an increase in lipopolysaccharide, which is a major component of gram-negative bacteria. So the authors thought, what could be causing these changes? What system in the body regulates so many others? the gut microbiome. So to look at the role of the gut microbiome, the authors had to look at the rat's poop, which doesn't look like this, it looks like that. And they looked at the rat's poop because the bacterial composition of poop is a way to tell what kinds of bacteria are present in the guts of animals. So when looking at the bacterial populations, the authors found that there was an increase in bacterial diversity overall in the alcohol drinking rat group, which was also identified in the more rare bacterial populations. And when looking at the specific types of bacteria in the gut, they found that there was decreased firmicutes and increased Bacteroidetes and proteobacteria. This is important to recognize because firmicutes play a role in calcium absorption, so less of them means less calcium being absorbed into the body. Bacteroidetes mature the immune system and increase inflammation, so by increasing these, more inflammation is occurring. And many proteobacteria are commonly found pathogens, which could all help to explain the nutrient level differences we covered earlier. To summarize all of the main points, the authors tested the effects of long-term drinking on rats, both young and old, and found, particularly in older rats, that they had decreased alcohol metabolism. But regardless of age, they showed increased signs of osteoporosis in the blood and morphological signs of osteoporosis. When looking at the cells in the bones, they found increased bone resorbing cell populations, increased inflammation, inflammatory factors, and inflammatory cells as well. And finally, they found that long-term alcohol consumption caused gut dysbiosis, which the authors hypothesize is actually driving all of the aforementioned change. So as a model, the authors speculated that increased alcohol in the body due to decreased alcohol metabolism can cause changes to the gut microbiota. The change of the gut microbiota activated T lymphocytes in the bone marrow to produce cytokines, thus affecting the proliferation of osteoclasts. Moreover, these effects may be closely related to age as the findings were exacerbated in older rats. Not only do I think that these correlations are exciting to investigate and learn about, they are also significant in a broader context. This information is significant because excessive alcohol consumption, number one, just has various harmful effects on many organs. And it also correlates with gut dysbiosis, which negatively impacts our immune system and bone metabolism. So by understanding the changes that take place, we can come up with ways to prevent these type of changes from occurring to reduce the incidence of disease. All science is basically a stepping stone for new knowledge, and these steps are driven by questions. And I had a few questions of my own when going through this information. The first question I had was whether these effects can be replicated in female rats, because throughout this study, only male rats were used to draw conclusions from. My second question has to do with whether there are other measures that can be taken in altering the gut microbiome to prevent the negative effects of alcohol on it and the rest of our bodies. And my third question is whether different levels of drinking can alter the outcomes seen in these rats. Will increased drinking or even binge drinking periodically increase the osteoporotic signals in the body? My final question has to do with you. What sort of ideas or questions popped into your head when hearing about this information? I would love to hear about it in the comment section below. Also, let me know if there are any topics that you would like to hear about in the future. Ultimately, I hope that you learned something, but more importantly, you enjoyed your time doing so. So if you did, give this video a like and subscribe for more in the future. That is everything for today. So thank you for watching, and I will see you next time.